It's a pleasure to introduce today Dr. Annemieke Aatsma-Rus, who will be giving the inaugural Fundación BBVA lecture. I think one of the most overused scientific terms in society in the last couple of years has been the RNA molecule. The COVID-19 pandemic has catalyzed the fair use in humans of vaccines based on the messenger RNA. However, what appears to be something new, at least for the society, has actually been used in laboratory by scientists for many years. That is the use of RNA as a therapeutic molecule to either direct the synthesis of a protein that is missing or defective in genetic disease, such as cystic fibrosis, metabolic disease, etc. We have the pleasure today of having an expert on this field. Annemieke she's professor of translational genetics at the Department of Human Genetics at the Leiden University Medical Center in Netherlands. Her work focused on developing antisense mediating exon skipping as a therapy for Duchenne muscular dystrophy or rare brain diseases. She has published more than 220 peer-reviewed articles, 11 book chapters, 15 patents. In fact, currently 2 Duchenne exon skipping drug has been approved for the use in human in the United States. But I would like to highlight her generosity. She has generated guidelines for the design of exon skipping antisense oligonucleotides for clinicians, for patients, for parents, for industry, and she is leading the first non-profit initiative for the design and development of individual treatment of RNA therapy for rare diseases. Thank you very much, Annemieke, for coming to Málaga, because you were in another Congress just a couple of days ago, and you have another one in 2 days. So thank you very much for coming to Málaga.
Thank you very much for the invitation and also for looking after me so well today. So what I was saying is that it's a pleasure to be here, and thank you very much for the invitation and also for looking after me so well today. And I don't know this society because I'm not from Spain, but from the talks that I've had with people that are involved, I get the impression that it's a really nice society and that people know each other quite well, and also that they're investing in the future, and I think that is quite good. So today I will talk about the Dutch Center for RNA Therapeutics, but I'll first go over my disclosure. So as Enrique already said, most of my career I've been working on Duchenne muscular dystrophy, and I'm still working on Duchenne muscular dystrophy, and my disclosures are all related to that, so they're not related to this talk, but for full transparency I'll go over them anyway. So Enrique mentioned patents, and based on these patents there's currently 3 exon skipping drugs approved for Duchenne in the USA. So my institute gets royalties, and I, as co-inventor, am entitled to a share of these royalties. All my other work, consulting, advisory boards, speaking at conferences, if there's a remuneration involved in that, that goes to my employer, so that goes to LUMC and not me personally. But as I said, not related to this talk. I'll give a brief introduction into what antisense oligonucleotides are for those who are not familiar. So they're short pieces of RNA that can bind specifically to transcripts. So they can be RNA or DNA analogs. They are chemically modified because, well, we need to give them drug properties. They have to be stable, they shouldn't be excreted. And as I said, they target the RNA, and they do this in a sequence-dependent manner with Watson-Crick base pairing. The aim, of course, if they're therapies, is to have a therapeutic effect, and they can have they can achieve 2 things. So either they can reduce the production of a toxic protein, or they can restore the production of a missing protein. So both applications work. And depending on your field, antisense oligonucleotides or ASOs are either small if you're working in the gene therapy or the cell therapy field, or they're big if you work in the small chemical drug field. So I mentioned already these antisense oligonucleotides, these ASOs, target transcripts, and they do this with Watson-Crick base pairing, and that's why they are very specific, and you can really get targeted effects. You can target transcript isoforms. So the side effects, unlike small chemical drugs, the side effects are not driven because they also target slightly similar proteins or enzymes. The side effects are mainly driven because of the chemical modifications that we give the ASOs to give them the drug-like properties. And of course, when you design your ASO, you can make sure that they only target a very specific transcript and not something else. And this really allows you to intervene at a genetic level. So not gene, it's not targeting the DNA, it's targeting the gene transcript, so the RNA, which means that the effects are transient, but it also means that a lot of the ethical issues that you would be dealing with when you do gene addition therapy or genome editing, you don't have here because you're targeting the RNA. Currently there is, well, depending on how you define them, there's 15 to 17 oligonucleotide therapies approved by the FDA and the EMA. I think the last one, the vaccines, probably most of you here have received the RNA vaccines from Moderna and Pfizer. And these vaccines, well, they're not really a therapy, they prevent that you get sick, but they do involve oligonucleotides. So they involve small pieces of RNA that code for the protein that then induces an immune response which protects you from COVID-19 infection. So those are protein restoration. I mentioned already, you can also use them to reduce the production of a toxic protein. There's a number of ASOs approved for that, mainly for liver diseases and for eye diseases. And then there's the splice switching oligonucleotides, which will be the focus of this talk, and these restore protein. And there's 4 approved for Duchenne muscular dystrophy. We're not going to focus on those. We're going to focus on nusinersen, which is approved for spinal muscular atrophy. And spinal muscular atrophy is a severe disease where children, if they're not treated, generally die before the age of 2, and the most severe type patients never manage to sit up. They die from respiratory failure. So it's really a very severe disease. It's a disease of the motor neurons, and nusinersen can restore the missing protein or increase the production of the missing protein, and this has quite a prominent effect if you treat pre-symptomatically. It seems that most patients are developing normal or almost normal. If you treat post-symptomatically, they're not behaving as normal, but they gain functions that they otherwise never would have gained, and the progression in later stage patients is slower. So this really is an example of what can be achieved with antisense oligonucleotide therapies. So why is this important, and why am I focusing on nusinersen? Because for the rest of the talk, we're going to see a look into the opportunities of this exon skipping approach. So I worked on muscle diseases, and delivery of ASOs to muscle is extremely difficult. And I mentioned these 4 approved drugs. They're approved based on minimal dystrophin restorations, where the question really is this enough to slow down disease progression? When we look at delivery to the central nervous system, if you treat systemically, the ASOs will not reach the central nervous system. However, if you do intrathecal delivery, the ASOs are taken up very well by the neurons and distributed throughout the central nervous system. So that now gives you an opportunity to treat locally, which means you need lower doses because, well, you have local treatment. It also means that the frequency is quite low. I mean, weekly intrathecal injections, no one is happy with that. But we know from nusinersen that you probably have to treat 2 to 3 times per year. And also another organ that you can do local treatment is the eye. So there you need to treat once or twice per year. And again, injections into the eye are not fun, but if the alternative is getting blind, then you're willing to undergo them. So these opportunities allow you to use very low doses, which means the systemic exposure is low. So the side effects that normally you get if you do systemic treatment, the risk of that is much lower because hardly anything ends up in the liver and the kidney, and locally you get very high exposure. And I mentioned already with low treatment frequency. Now there's one type of mutation that is extremely eligible for exon skipping, and that's the cryptic splicing mutation. And I'll briefly explain that. So this is gene, and of course the coding domains are located in the exon, and in between you have the non-coding introns. During transcription you get a copy, during splicing all the exons are joined together, and that is then translated into protein. So that is basic biology. A cryptic splicing mutation is a mutation where part of the intron, due to the mutation, is recognized as an exon and then ends up in the messenger RNA. And that prevents translation into protein, because either the cryptic exon is out of frame, or it contains a stop, or it contains amino acid that disrupt protein function. But these cryptic exons prevent translation into protein. If you use ASOs to target these cryptic exons, what happens is that you restore normal splicing. And the nice thing then is that you restore the normal protein. So the problem that we have with dystrophin exon skipping, if you're familiar with that, is that we restore a partially functional protein. And then the question always is, how functional is this protein? Here you would be restoring a normal protein, so you don't have that question, because obviously the normal protein is functional. So these mutations, if they happen in eye or brain diseases, are really extremely attractive cases, because you can do local treatment, you can restore the normal protein. However, these mutations are very rare, and that makes them very unattractive to industry, because there's no commercial value if you develop something for 1 or 2 patients. So the numbers are too low. And then one of the questions that Tim Yu in Boston Children's Medical School asked himself, well, can we do this in an academic setting? And he actually identified a cryptic splicing mutation in Mila, this a young girl here, and she had Batten disease. And on one of her alleles, she had a cryptic splicing mutation. And he discovered this shortly after nusinersen was approved. So he realized, well, I could in theory treat this patient if I had an ASO targeting this cryptic splicing mutation. So he was developing it, and Mila's mother was willing to raise funding to actually facilitate this. So after the genetic diagnosis, the lab designed the ASOs, they tested them in fibroblasts. He had discussions with the Food and Drug Administration, so what type of toxicity studies should I be doing? So they did rat toxicity studies, and then he applied for an investigational new drug. And within a year after diagnosing the patients, he did the first treatment intrathecally with this ASO that he called Milasen, after Mila, the patient. Then of course the question is, did this have a therapeutic effect? And this is a slide based on a seizure diary that Mila's mother, Julia, was keeping. So before the treatment was started, Mila would have a lot of epileptic seizure, and that's part of the Batten disease. So it's a progressive neuro-wasting disease, but it's also associated with a lot of epileptic seizures that don't respond well to anti-epileptic treatment. So before the Milasen treatment started, usually Mila had 20 to 30 epileptic seizures per day that lasted more than a minute. And then you can see here the treatment started first at a low dose and then increasing doses. And you can see that the dots become lighter, so the seizures lasted a lot less, usually a few seconds. But you can also see that generally the dots are lower, so she had less seizures per day. So that is quite clear a treatment effect. However, sadly, by the time Mila was diagnosed, there was a long diagnostic odyssey, because she had this cryptic splicing mutation that was difficult to discover. So she had accumulated already a lot of irreversible neuronal damage, and sadly she passed away last year. So this treatment was not a cure. Of course, everyone is wondering, well, what would have happened if she had been treated earlier, if she had been treated when she was 2, 3, 4 years old rather than 7 years old? Well, we'll never know that. What we do know is that the parents of Mila say the fact that she had this huge reduction in the seizure frequency and duration, for them that improved their quality of life tremendously. So for them the treatment was a success, even though sadly Mila passed away. Well, when we heard this story in the Netherlands, we thought, well, actually, I mean, this is something that we should be able to do as well. Because we have also these cases where we think, well, these patients are very eligible for exon skipping. Normally, when we develop exon skipping, at some point, we hand over to the pharmaceutical industry, and then they develop them clinically. they're not interested in these small groups. So can we use our expertise in exon skipping therapy development to do the same thing that Tim Yu did in the US? Can we do this in the Netherlands? So we teamed up. So I'm not a brain person, I'm a muscle person, but I did develop these guidelines that Tim Yu actually used to develop Milazen. My partner in crime at the LUMC is Willeke van Roomom. We've been sharing an office for more than 15 years. She works on brain and also on exon skipping. We also teamed up with someone from Radboud in Nijmegen, Rob Collins. He is someone who's been developing ASOs for eye diseases. And we teamed up with Ipe Elgersma from Rotterdam Erasmus Medical Center because he's working on ASO development for neurodevelopmental disorders. So we cover the whole spectrum, and our aim is to do what Tim Yu was doing. Can we develop these ASOs for very rare cases within an academic setting? So it's not for profit, we don't want to make any money, we want to develop this and then treat the patients free of charge. So for whom is this? So mutations must be very rare. We're not going to compete with industry because we don't have the expertise to do that. So really the unique or close to unique cases. We work only on exon skipping. There's other approaches, so you can do the knockdown of toxic proteins. We have no expertise in that, so we're not developing that. We're only developing exon skipping. The target tissue has to be the brain or the eye, so we can do local treatment and keep the cost relatively low. And very important, the patients should have benefit from the treatment. So you can look at it in two ways. So first of all, if the mutation causes all of the problems during embryonic development, treating postnatally won't have any effect. So these patients would not be eligible. Also, once someone is gone fully blind, restoring the missing protein will not make them see again. So then it's too late. However, if patients, even if they're in a very late stage of disease, so if they're legally blind but they can see a little bit, for them that last bit of vision is of course incredibly important. So those patients would be eligible for the treatment. And obviously the patients must be willing to undergo such a treatment. There's no guarantee that it will work. There might be safety issues as well. So there is a risk, and so this really is something that we have to co-develop with the patients, because if they don't want it, if they don't want to take the risks, if they don't think the burden of injections into either the spinal cord or the eye is worth the potential benefit, then obviously it stops. So we launched ourselves February 29, 2020. So February 29 is Rare Disease Day. We thought that was a very nice day to start. We had big plans, but of course, shortly after our launch, COVID happened. So the lab was closed and we couldn't do any experiments. But fortunately, what we could do, and I'm sure you are all aware of this, we could have Zoom meetings with a lot of people and agencies and institutions. So we had interactions with the European Medicine Agency, with the Dutch, so the Medicine Evaluation Board, the Dutch EMA, Zorginstituut Nederland, that is the Dutch HTA technology, so the payer system, patient and patient organizations, our hospital pharmacy, the Ministry of Health, and many others. And really we just had discussions like, this is our plan, this is what we want, how are we going to do this, and how will this, well, is it possible to do this and then be reimbursed? What are the procedures? Because we're doing something that's not been done before, and of course the drug development path and processes, they have not been set up for treating single patients. They're always set up to treat larger groups of patients. So we're really inventing the wheel within Europe. So what we've discovered is that things are different between Europe and the US. So in the US, if you want to do this, you need FDA approval even to treat a single patient. So you need to show them data that this is effective and safe. In Europe, you don't need that. So in Europe, a clinician can treat a patient in a named patient setting, which means that the clinician takes full responsibility and says, okay, based on preclinical data or based on literature, I think this compound, this drug, is going to be beneficial for you and it's going to be safe. The nice thing is that you don't need regulatory approval for this. You need approval from the hospital inspection. So that means our logistic and administrative burden is a lot less than it is in the US. So we don't need regulatory approval, but we are communicating with the regulators for advice. Because of course we do want to do this in a safe way. We don't want to make things worse. So what we've learned from all that interaction is that we don't need GMP, so good medical practice compounds. And this is very important for two reasons. One is costs, because if you want GMP product, it's really expensive. But it's also the waiting time. So if you want a GMP production, that's often like a year to 1.5 year waiting time. Well, we aim to treat progressive patients, so 2 years means they have irreversibly lost one or two or more function. And it's also important because if you do GMP production, so normally this is done at ton scales, 1,000 kilograms, and then you may need 100 grams to do all the analytics. If we have 10 grams of ASO, we have enough to treat a patient for 100 years. So then we would have to make 10 times more of what we need just to do all the analytics. So we're talking with the manufacturers and we're talking with the regulators, like how can we do this in such a way that it is safe, but that it's also quicker and we don't waste, well, 90% of our compound on analyzing it. Another thing is that we can do our preclinical studies and safety studies in-house in a GLP-like setting. And GLP-like is not, we try to do it as good as we can. GLP-like really means that we do it under GLP settings. We don't have the certificate for GLP, but we do adhere to a lot of the rules that should be done in a GLP setting. Again here, if you do it in a GLP-like setting in-house, the costs are 20 times less than if you would do it in a certified lab. And again, also the waiting time is about 10 months less. So we want to keep the cost as low as possible. And then what's also important, if you treat one patient, there is no placebo. There's before and there's after. So when a patient has been identified as he or she is eligible for this, we start discussing with the patient, so what would be benefit to you? And how can we measure that benefit? And then collect natural history of the patient while we do all the in vitro optimization, the in vivo safety studies. So we have this natural history, we have the trajectory, and then we can compare before and after treatment. And of course with Mila it was really clear because there were epileptic seizures and you could see a reduction. But if you have a slowly progressive disease where you can measure something with MRI over the course of 2 or 3 years, it is more complicated. So we have a preliminary timeline, and I'll admit, so we've only been in this first part. So the testing of the ASOs, seeing whether they're efficient, the functional study, so that means is the missing protein back, if there's an enzymatic deficit, is that restored? All of that we're used to doing also for when we want to treat larger groups of patients. So we're fairly confident that we can do this for about 100,000 euros in about 12 months. Then the uncharted waters is this part. So the GMP-like production, the safety studies, how will that happen in-house? So we estimate, based on all the information that we have now, that this will probably cost us about 500,000 euros, but we've never done it, so we don't know for sure. And then of course there's maintenance costs because patients will be treated 4 to 6 times per year, maybe more, maybe less, depending on the disease and how things are developed. And they have to be monitored. And as I mentioned already, the natural history collection really starts already early on because you don't want to measure what happens after treatment, you want to compare before and after treatment. So this is a graphic depiction of what I just said. So we'll start with patient-derived cells. If we can use fibroblasts, we're going to use fibroblasts because they're much easier to work with than iPS cells. But sometimes if the transcript is only produced in neuronal cells, we'll have to use either transdifferentiated fibroblasts or iPS cells, and we'll use them. And another thing that we'll try, so for now we want to do also safety studies in rats, but if we find something that's toxic, we want to see whether our neuronal iPS cells are predictive. Because if they are, maybe we can skip the in vivo safety studies and use in vitro instead. But that is something that we're working towards. So we're currently working on 2 different diseases. One is called BPAN, beta propeller associated neuropathy. That's an iron accumulation disorder, and we have identified 2 patients with mutations that are likely eligible for exon skipping. We have the ASOs to do the exon skipping, and what we're currently studying is whether the missing protein is restored and whether that leads to functional effects. And what we have here, so we have 1 mutation that's within an exon, but we think if we skip that exon, the resulting protein is still functional, but that's of course something that we need to confirm. The other one is a cryptic splicing mutation, but it's quite a tricky cryptic splicing mutation. So we see that the cryptic exon is removed, but we don't know whether other things are happening. So we really need to show that the protein is restored. And in Nijmegen, they're working on a patient with Stargardt disease who has a cryptic splicing mutation. And they've shown before that they can manage to skip this exon and restore the missing protein. However, their current ASO that they have is not very efficient. So they're now working on optimizing the ASOs for lead candidate selection, and they're already working with the patient to see, well, how can we measure vision in this patient and loss of vision before and after treatment. Now all this was in the Netherlands, and of course when Tim Yu presented the Milasen story and when it came out in the New England Journal of Medicine, lots of people thought, well, this is something that we can do, or this is something that we want to do because we want to help the patients. And I'm part of the Oligonucleotide Therapeutics Society, and as a society we saw a huge opportunity for these individual cases that otherwise never would be treated, but we also saw a risk. Because, well, we have 20 plus years of ASO development expertise, but there's also people that have less expertise. And that based on the story of Milasen can think, well, if Tim Yu managed to do it in a year, how difficult can it be? Well, there's lots of considerations and things. So we said we need to provide guidance to people. So we were planning for a stakeholder meeting, again pre-COVID, we were planning for a stakeholder meeting in April 2022. Obviously that didn't happen. It also didn't happen online because FDA was a big part of our meeting and they had other things on their minds at that point. But what we did do, we produced a briefing document. So we were going to produce this anyway for all participants, like this is the current state of the art. This is what you need to know about the efficacy, the safety, how do you design, how do you test. So we produced that briefing document and we put it online. So if you want to see it, you can scan it, or you can go to the website of the Oligonucleotide Therapeutics Society. And the group of people that was involved in these efforts, trying to have this meeting, producing this briefing document, now has also formed the N of 1 Collaborative. So that's a global umbrella organization. So the current landscape, if you base it from the European or Dutch perspective, we have in the Netherlands the Dutch Center for RNA Therapeutics. Within Europe, we have the One Mutation, One Medicine initiative that also tries to work towards this. And globally, you have the N of 1 Collaborative. And all of them have the same goals. However, different jurisdictions have different rules, so that's something to figure out. And we try to help each other where we can, but sometimes there's problems that are, well, countrywide or continent-wide. So introducing you to the team of the 1M1M initiative. So this is led by, again, 2 people that you already know, and 2 clinicians and a project manager from Tübingen University. And they're involved in the Rare Neurological Disease European Reference Network, and they have a big network. So together we're trying to identify eligible patients and to provide guidelines and input for clinicians who want to be involved in this. Then we have this global umbrella organization, the N of 1 Collaborative, and this really is something that's open for everyone in this field. And what we want alignment and transparency. So we want everyone to learn from successes, but also from failures. Because when we are going to do this, not every story will be a success story, but we can probably learn more from the failures. We can learn a lot from the ASOs that turn out to be toxic. So we want to provide guidelines, and we're working hard on this. We want to share safety and toxicity data. We want to provide a clinical toolkit, because the N of 1 treatment, the ASOs, you need to develop the ASO for one patient, but you also need to develop the outcome measures for one patient. So how to measure in one patient that this works, you need to customize that as well. So we will provide protocols, best practices and procedures and registries. And like I said, the different subgroups provide different things and then we share everything. So it's really an open and collaborative effort. So within the 1M1M network, so I'll now show different slide formats to show you who was responsible for what. So we had a lot of discussions between preclinical geneticists and clinical scientists about how to select the patients, because it's not just they need to have a mutation where you can skip the exon. There's much more to it, because so the variant needs to be correct, but also the disease, it needs to be a progressive disease. If it's a stable disease, how will you measure an effect? If it's a disease where all the damage is done in utero, treatment is not going to help. So you really need, the disease needs to be treatable, so brain and eye and progressive diseases. And then the patient also needs to be fit, say the criteria, because if the disease stage is too advanced, it's too late. But also if the patient doesn't want to be treated, then it's not, he or she is not eligible. So we had one case of a patient who was very eligible, ticked all the boxes, but the patient was incredibly afraid of needles and did not want to have intrathecal injections. Well, if that is his argument, then of course we're not going to develop a treatment and force it on him. So all these things have to fit. And then if you see, well, which diseases are eligible, well, basically what it comes down to, which genes, which mutated genes are eligible. So at the DCRT we have now a list of 700 genes where we really check, like, these lead to progressive neurodegenerative diseases. So they can be found on the website of the DCRT, and we're currently expanding it, because so there's these 700 were the easy, the obvious choices, but there's more where you say, well, maybe sometimes this is also eligible. So we're working on that. Then seeing whether something is eligible, I mentioned for cryptic splicing mutations it's easy because, well, you have part of an intron that suddenly is within the mRNA, and if you skip it, everything is back to normal. It's not that simple, because based on where the cryptic splicing mutation is, sometimes what seems eligible is not eligible. And that is, say, the conclusion of this large scheme. If you want to skip a mutation with an exon to bypass a mutation and to still allow production of a partially functional protein, it's even more complicated. And currently we're doing all these things by hand, and, well, depending on how likely it is that the mutation is eligible, this can take anywhere from 5 minutes to more than a day. So we're developing now software tools to help us with these processes, because a lot of things are or can be automated. So we're working on that. And then we also had input from the regulator. So we went to the regulators as the DCRT, but also as the 1M1M consortium, so the European regulators, what is their perspective? And they were very clear about one thing, and that is that if you cannot measure a therapeutic effect that is relevant to the patient within a time frame of 2 to 4 years, it's not justified to start a treatment. So if you can only measure 10 years down the line, that is too late. The burden of intrathecal treatment is too high, especially for severe progressive diseases where the burden of the disease is also already high. So you really need to have outcome measures that meet the criteria. So it needs to be, well, testing something for the disease stage of the patient. So even if you have 2 patients with the same disease, and one is in an early stage and one is in a later state, likely you will need different outcome measures for each of these patients. They have to be individually meaningful. So the benefit, so a quote from the Dutch Duchenne chair of the Dutch Duchenne Parent Project, clinically relevant doesn't mean what is relevant to the clinician, it means what is relevant to the patient. So that is something that we really have to bear in mind. Is it meaningful to the patient? And then of course it needs to be sensitive. So can you measure before versus after treatment? And with the epileptic seizures going down, that was relatively straightforward, but sometimes it's not that easy. And also, can you do it inter-individual? So knowing the natural history, if you know that all patients would have lost ambulation, for instance, at age 15, and your patient is still ambulant after treatment at age 17, well, that is quite clear a difference from the natural history. And mentioned already, this needs to be done in a reasonable time frame and obviously responsive to the treatment. So if you want to read more about this, we wrote a paper about this as well as the 1M1M consortium, and not just the patient eligibility criteria, but also all the ethical concerns, and we've not gone into them, but of course there's a lot of ethical concerns related to this as well. So then what is another problem that we face? That is that cryptic splicing mutations are rare, or that's what everyone believes. But the problem is, usually when we look for mutations, we do exome sequencing. Exome sequencing is not going to tell you that there's a cryptic splicing mutation, because these are in the introns. So the problem is that many people only look at exons and we don't find the intronic mutations. Well, these intronic mutations would be very eligible for treatment. So if you have a cryptic splicing mutation in a progressive brain or eye diseases, by all means do email us if you're interested in ASO development, because we can link you through DCRT or the 1M1M network to relevant people. And then finally, a problem that is more global is that the drug development for normal drugs is not fit for purpose for N-of-1 treatment development. There is flexibility possible both with the EMA and the FDA, so they do see the problem and they don't impose all the regulatory things that apply to the common disease or the more common rare diseases. But the question is, where does the flexibility end? And it looks like in Europe, everything is possible for N-of-1. You could even forego toxicity studies in animals and just go into the patient directly, provided you use the nusinersen chemistry. But as soon as you would do it for 2 patients, you would have to do GLP compliant safety studies. So that is very tricky, especially if you're already treating 1 patient and now you're treating a 2nd patient, suddenly you have to do rat safety study. Well, you know the safety in 1 human with the disease already. So that's a discussion that we need to have with the regulators, but we plan to have it when this becomes a concrete problem rather than a theoretical problem. The FDA is more flexible, so for them N-of-1, 2, 3, 4 doesn't really seem to matter. And well, as someone said, depending on the day, N-of-1 can mean anything between 5 or 15. So there's more flexibility there, but it's also not strict. So sometimes 5 is okay, sometimes 10 is okay. But the question is, of course, because at some point, if there will be a threshold, what about these patients that have slightly more frequent mutation, where normal drug development is still not going to work because it's too small, companies are not interested, but it's too big for N-of-1? Well, these patients are, well, still missing the boat, as we say in Dutch. What about geographic variability? And I think why the regulators are quite strict is what they don't want is that we develop something in an N-of-1 setting, and then it turns out that there's 200,000 patients in China or Brazil that also have this mutation and then can, without doing any proper development, can get this drug. That is, of course, not what we want, but that is probably why they are afraid and a bit cautious. And also what we want to do is we want to balance speed, efficiency, and safety. And I like to use the example of the ambulance. Because these diseases are progressive, we know that, well, the longer we take, the more function the patients will lose. But on the other hand, there's also ways that we can make it worse. So if you use the example of the ambulance can speed, the ambulance can go through red lights. But what you don't want is that the ambulance crashes or drives into a wall. You don't want that. So we really have to very carefully balance safety with also speed. What we've discovered is that transporting patients is easier than transporting ASOs, and that may seem counterintuitive. So we thought very naively that if you have an ASO for a single patient in the US, and it turns out that there's a second patient in Germany who would be eligible for the same ASO because he has the same mutation, that you could just ship the ASO. But that turns out not to be the case, because this ASO, of course, it's an FDA-approved drug, because you need this IND. So that means you can't export the ASO without a lot of administration and difficulty. So it's actually easier for the patient to move to the US with his mother, to live there for a year, be treated, and then once the treatment is established and initiated, the patient and the ASO and the mother can go back, be reunited with the rest of the family, and then the treatment can continue in Germany. And if I say it like that, it sounds like there must be a way around this. So this is something that, again, we also want to discuss with the regulations. Like, we understand why there's regulations, and we understand why you can't just transport drugs here, there, and everywhere, but for these rare cases, hopefully exceptions can be made. And the final challenge is that because we do it in Europe on a named patient basis, this really relies on a clinician willing to champion this and because he or she has full responsibility for the treatment, but also the administrative burden of applying for the hospital inspection approval, the monitoring, etc. If you want to know more, so this is our website, I mentioned already, rnatherapy.nl, so you can find a lot of information on there. And in summary, I think, or I hope I've shown you that ASOs offer unique opportunities for mutation-specific treatments. N-of-1 development is possible for brain diseases. That has been shown in the US now multiple times. For eye diseases, it's not been shown, but we have approved ASOs for the eye, so it should be, in theory, possible as well. The context of each individual case is different, and also the context, the jurisdiction can lead to different things. However, there are common processes, and for that we're trying to work together nationally, European-wide, and also globally to really align and facilitate as much as possible. And I think it's also clear this needs multi-stakeholder improvement. So we need clinicians, we need preclinical scientists, we also need regulators, ethicists, and obviously the patients, because they really are involved in determining the risks that they are willing and unwilling to take, and the burdens that they're willing and unwilling to take, and the benefit that they would want in return. Final 3 slides. First, a shout out to the DARTER network. So this is a cost action, European-wide action on RNA therapy delivery. So not for N-of-1, for everyone who is working in the oligotherapy field. The action will run for about half a year more, and the nice thing about cost action is that until they end, additional people can join. So if you're interested, antisensRNA.eu, and you can still join if you're not already part. And I mentioned already the Oligonucleotide Therapeutic Society. Well, like this meeting, we'll have our first in-person meeting next month in Phoenix. It's our 20th anniversary, so we're a rather young field and a rather young society, but we're really looking forward to meeting everyone. So if you want to know more about oligotherapies, I invite you to attend. And then finally the acknowledgement slides, because I mentioned already, this really is a multi-stakeholder effort. So within each of the different centers at the DCRT, it's not just a group of preclinical scientists that think they can do it. We need the clinicians, we need the hospital pharmacies, we need the people from the animal facilities to do the GLP-like studies. So we really need all of them to work together. And this is a picture of the Leiden group that has expanded since we started in 2020 with 2 people. So with that, I thank you very much for your attention, and I'm very happy to discuss any and all questions or concerns or considerations that you might have.